Alrighty, hello everybody. Today we're going to be taking a look at Lecture 10, which is talking about Transcription 1 and RNA polymerases. But yeah, let's go ahead and get into this. Again, I think I'm posting these a bit late. Had my own exams. Talked about this in Lecture 8, or Lecture 9, sorry. But yeah. Lecture 9, we talked about, like, replication. I talked a lot about these two. And then, like, this. And I didn't really talk about this too much. This is something you probably should ask your professor more about. I'm sure if you came in and asked me in person, I would probably have more to know more about this. But at the time of recording, I'm a little bit rusty on that, so I'm not super confident in my knowledge on that stuff. But the flow of genetic information, we're going to talk about transcription, which is going from DNA to RNA. And yeah, let's go ahead and get into it. Definition of transcriptome. Very briefly put, it is the total amount of RNA in a cell. That's it. That RNA can be tRNA, rRNA, mRNA, whatever, right? And the RNA is always specific to a certain cell type. So different cells have different RNA contents and makeups. So there's different types of RNA. The ones you're going to be covering mainly are tRNA, rRNA, and mRNA. In 442, you guys might talk about microRNAs and silencing RNAs. And then very briefly, you'll cover maybe snRNAs and long non-coding RNAs. But definitely these three are the important ones. These three. And the one specifically that we're going to talk about for transcription and then especially translation in the future is mRNAs. So what you should know from this is, first of all, the largest amount of rRNA is actually, the type of RNA with the highest abundance is actually rRNA. rRNA is massive. It takes up so much of the total RNA content of a transcriptome, and that's just because you have millions of ribosomes, right? It should make sense, right, that if you have a lot of proteins that need to be translated, you need tons and tons of ribosomes. And ribosomes use rRNA to make their structures along with our proteins. The ribosomal proteins. So our ribosomes are made of rRNA and ribosomal proteins. That's where most of your RNA is from. The other part is from tRNAs, lots of tRNAs, right? They're around 70 to 100 base pairs. Please memorize these numbers. But yeah, there's quite a lot of them. Same thing here, 10 to the second, 10 to the third, so 100 to 1,000 base pairs for rRNAs. And then for the mRNAs, there's a decent amount of them, but they make up like 5% of the total transcriptome. They're also very diverse. That's the other thing. rRNAs and tRNAs, not very diverse, right? There's only so many tRNAs you can make because there's like 20 amino acids, right? And like 61 codons, or 64 codons technically. But it's only 61 of them are actually tRNAs, if I'm not mistaken. And then because for the 3 stop codons tRNAs do not bind to stop codons. Release factors do. Release factors are proteins, right? But for the mRNAs code for a bunch of proteins, right? There's alternative splicing and all that jazz, a whole bunch of stuff, right? And because of that, right, they're very high in diversity, but there's not a lot of them. Here's just estimated composition. rRNAs 90%, tRNAs 10 to 15, mRNA 3 to 7. Cool. Not too interested in that. This is just a nice way of showing you that different cells have different genes that are being expressed. I hope that's something that you understand by now, right? Different cells should not be expressing the same genes, or at least they should not be expressing the exact same genes, right? Maybe a neuron probably needs more, let's see here. A muscle cell, right, is going to express stuff like actin and myosin so it can do cross-bridge cycling, right? But a neuron doesn't need to do that. A neuron doesn't need to contract. So it's not going to express proteins or express genes that code for like actin and myosin quite as much as a skeletal muscles, right? So just something to keep in mind. And this kind of shows, if you had different genes, right? Different cells, like maybe, so this neuron requires this a lot, right? It's like a cardiomyocyte. Maybe this gene, right, codes for actin, or codes for myosin, right? The neuron doesn't really need myosin, and then a white blood cell certainly probably doesn't need myosin, right? So, you know, stuff like that. Although lymphocytes might actually use myosin for movement, but I digress, right? Basically, different cells have different transcriptomes, right? Because different cells have different functions. This is just a nice way, a way of saying transcription is selective, right? With DNA replication, right, we replicate the whole genome. But with transcription, we only transcribe certain genes. Now we get into the structure of RNA polymerases. There's three different kind of domains that I want to talk about. It's the catalytic site, the core domain, and the mediator/interactive domain. And I believe I'll talk about those more in a second. So the first thing is the catalytic site. The catalytic site is this section here. It's where the reaction takes place. And I hope it makes sense that that region is conserved, because evolution and nature have pretty much optimized the catalytic site to be able to do that nucleophilic attack and chain formation, right? So, you know, the formation of an RNA molecule, right, uses like the same magnesium cofactors, the same aspartates, right? Similar to how DNA polymerase does, right? DNA polymerase uses the same thing. It has this NTP binding site, all that stuff. If you were to break that, right, the polymerase just doesn't work anymore. And so that's why it's highly conserved across organisms. The core domain, right, is relatively conserved. It's conserved a decent amount, right? It's this section out here. It's basically what helps with NTP incorporation during chain elongation. So basically, it takes the NTP from the cytoplasm, if you will, or in eukaryotes' case, from the nucleus, and it takes the NTPs from the cytoplasm and helps the catalytic site incorporate them. Relatively conserved, but not as conserved as the catalytic site. The last part is the mediator/interactive domain. So that's the final outer portion here. And this portion is highly variable. And it's highly variable, right, because different organisms have different transcription factors and different binding proteins that assist different RNA polymerases with binding to DNA to perform transcription. So, you know, to reiterate that, different organisms have different proteins, right, that regulate transcription. And because of that, this mediator/interactive domain at the very edge here needs to be very malleable and have the ability to change very easily, because, you know, we can't just take a polymerase from a human and stick it into a bacteria, because there's the interactive domain for a human is very different from a bacteria. So this area is not as conserved as you know, something like the catalytic site or the core domain. Here is a diagram of the mechanism. I'm not going to go over this again. I feel like I've covered this enough at this point, but hopefully you recognize, right, the O- attack, right, the 3' attack to the 5' phosphate, the 5' triphosphate, releasing a pyrophosphate, which can be cleaved for energy, and then the usage of these magnesium ions to stabilize the negative charges of these oxygens, right? And then the aspartates in the protein to help, aspartates in the RNA polymerase to help kind of stabilize those magnesiums, if you will. What you should memorize is, one, this catalytic mechanism is located in the beta-beta prime subunit, or the beta-beta subunit. Copy this down on your note card, memorize it. The catalytic mechanism is very similar to DNA polymerase. You really shouldn't have to memorize anything extra than what you already had to for DNA polymerase. Similar to DNA polymerase, 5' to 3' synthesis, right? And then, of course, we have the 3' hydroxyl that attacks the alpha phosphorus, right? The first phosphorus of an incoming NTP. We use magnesium ions, right, to stabilize the negative charge, and they're very efficient, right? Very fast. So in E. coli, 15 to 90, yeast, 20 to 70, and this TAC7 polymerase is a bit different, but this one's ridiculously fast at 200 to 260 nucleotides per second. The core domain is made up of 5 different subunits. It's got the alpha, the 2 alpha subunits. It's got the beta and beta prime from the catalytic domain, and it's got the omega subunit, right? I wouldn't worry about this number here. I believe this is the mass, if I'm not mistaken. But what you should know is this is the region that would have any kind of proofreading stuff. So, firstly, it lacks a proofreading thing for the 3' to 5' exonuclease. So, no 3' to 5' exonuclease. It makes errors, not super often, but it does make errors. And then the polymerase footprint encompasses 35 base pairs of DNA. So, that would be like this here. Like, that's the footprint, if you want to think about it like that. It's like covered by the polymerase, right? And then like this area here is the footprint, basically. So 35 base pairs are basically like stuck inside the polymerase at a time, and 17 of those base pairs are actually unwound, which is like this section right here specifically, like the section here that's split. This is unwound. So to memorize things from this, I would say memorize the fact, memorize this 5 core subunits. Memorize that it doesn't have a 3' to 5' exonuclease. Memorize that it has a rate, like some error rate, not too crazy, but it does. And then I would say memorize this. Memorize that it encompasses 35 base pairs and that 17 of them are unwound. For the final one, we have the mediator/interactor. Honestly, you shouldn't have to memorize any of these. What you should know, though, is that this is very nonspecific. It changes from organism to organism, and there's a whole bunch of different subunits that bind to different DNA sequences. So there's a whole bunch of random stuff. So like this sigma subunit is an example of one of those. So the RNA polymerase holoenzyme. Remember, the holoenzyme is a combination of enzymes that makes up a whole. Comprises of a variable sigma subunit. So we got a sigma. You should hopefully remember sigma from Bio 200. We talked about sigma a little bit there. And that sigma binds to different DNA sequences at the promoters, right, to cause transcription. So there's all sorts of different sigmas that you could use. And yeah, that's an important thing to keep in mind. So for eukaryotic RNA polymerases, it has a similar layer of stuff. So in prokaryotes are very simple because they just have sigma. Sigma binds to the promoter, and the promoter binds to RNA polymerase, and that helps with transcription. And yeah, there's many different types of sigmas, and these different sigmas are involved in different types of cellular functions, like housekeeping, nitrogen metabolism, stationary phase, heat shock, whatever. There's a whole bunch of stuff, right? So, and they also have different binding sites, but basically everything goes through sigma in prokaryotes. Eukaryotes, though, are weird. So eukaryotes, they use RNA polymerase 2 specifically, right? Memorize that. It's specifically RNA polymerase 2. And it has a similar layer of structure. But what you should know is here there are 12 subunits. There are 12 subunits in the core enzyme. Don't have to know specifically what they are. But the catalytic domain is RBP1 and RBP2, and they have high homology to the beta-beta prime from the RNA polymerases in prokaryotes. Also, just to keep in mind, these polymerases that we talk about up here, generally speaking, we're talking about prokaryotes, right? Although they do apply to eukaryotes as well. Now, in eukaryotes, for prokaryotes we just have sigma, right? But in eukaryotes we have 7 general transcription factors. And these, I would memorize these. So TF2A, TFB, D, E, F, H, and TBP. This TBP stands for tautile binding protein, something you probably already also saw in Bio 200. There's also a core enzyme So these all bind together to form this pre-initiation complex that is the pre-initiation step for translation. And this PIC needs to complex with a multi-subunit mediator complex for normal transcription. So there's some basal rate of transcription, right, if you just have the 7 transcription factors, right? So maybe if you have 4 of them, right, it's not going to be the basal rate. If you have 7 of them, it's going to be the basal rate. But then if you want, like, upregulated transcription, you need the mediator as well. And specific TFs regulate transcription factor mediator interaction. So these are general transcription factors. You need these for transcription generally, but there's also gene-specific transcription factors as well. So there's transcription factors outside of these 7 here that help with specific genes. All this to say, like, is that, if you have these 7, you're going to produce transcription at a certain rate. But then if you add the mediator complex, it's going to be significantly faster. And then if you add any more gene-specific transcription factors, it's going to be even quicker, right? So basically, the more transcription factors and mediator complex you add, the faster it gets. Here we have just an example of what transcription looks like in yeast and E. coli. So yeast eukaryotes, right? And then E. coli prokaryotes. So see here we have a promoter. We have our sigma that binds to RNA polymerase, right? And then it goes around. You initiate, you terminate, you elongate, all that stuff, right? In eukaryotes, very similar, right? We start here, right? We have all of our transcription factors. They all bind together. We do all this, right? We elongate, do all that, and then we produce our transcription, our RNA. So, viral T7, so this is TAC7, if I believe. I think it's called TAC7, if I'm not mistaken. TAC7 polymerase is a 1 subunit RNA polymerase, and it binds to a single promoter. It is a bacteriophage RNA polymerase, and it is extremely efficient. It is like twice as fast as most other polymerases. So, what you should know about this is that it's highly specific to a single promoter. So it won't bind to anything else. So it's very useful for experiments, right? If you want to transcribe a certain gene, right, you can just stick this thing in there, make the promoter of the gene of interest the promoter for this specific polymerase, and then you can just make a bunch of copies of that RNA. It's structurally similar to DNA polymerase and reverse transcriptase, and like I said before, it's single subunit. Other than that, it's very quick, right? It's helpful for making mRNA vaccines, right? Because, mRNA vaccines are literally just you producing crap tons of RNA, right? And this thing produces RNA very quickly, right? And then it should make sense, right? To do the reaction for any polymerase, you need NTP and magnesium, right? Because magnesium is a cofactor for the active site, and NTPs, well, you need NTPs to make RNA, so, yeah. And here is just a nice schematic of, like, what you should be thinking about when, for the different RNA polymerase structures, so. We have our T7, right, polymerase, which is 1 subunit, binds 1 promoter. We have E. coli, right, many different transcription factors that bind. There's a bunch of different sigmas, right, and then the 5 core subunit, the alpha 2, beta prime omega, right? So 2 alphas, right, a beta and a beta prime and an omega, that's 5 there. But then for eukaryotes, we've got 12 different subunits. We've got our 7 general transcription factors. And then we have, a mediator, a mediator complex of some sort, and then we have a bunch of extra subunits, right? And then, of course, we have any transcription factors that are gene-specific and stuff like that. So, yeah. The RNA polymerase evolution strategy to achieve, like, this higher multi-specificity that it's talking about is, firstly, keeping this site conserved, right? We don't need to change the catalytic site if it already works. So no need to change that. But what we do change, right, is the less conserved region that binds to different transcription factors. So the way that it achieves this specificity, right, is through its ability to bind in the less conserved regions to different transcription factors. And these transcription factors, they only show up in the cell at certain times, right? So maybe if the cell is under stress, certain transcription factors will show up, and if those transcription factors show up, they will bind to RNA polymerases and produce more RNAs. But basically, the evolution strategy for this is that, RNA polymerase transcription, the transcription for RNA polymerase depends on environmental signals. And when it says that easier to change to increase regulation complexity, yeah, you can just change the conditions of the cell, right? So just change what the conditions of the cell are, and then as a result, you'll make different proteins, and those proteins will bind to the polymerase in different ways, right? And cause different transcriptions. So that all occurs in this less conserved region, right? In this outer general TF and mediator binding area. So this is ChIP-seq. Very briefly, I'm not going to talk about this too much. Basically what's going on, you talk about this a lot more in 442. Basically what's going on is if I've got a section of DNA, right, that's maybe a promoter region, right, or like a enhancer region, or like a transcription factor binding site, right, and I want to figure out what that binding site's region is, like what the DNA is there, right. What I can do is I can wait for the transcription factors to bind to the DNA, right, and then what I can do is I can basically solidify the DNA and the protein. So like the protein and the DNA will become covalently bonded and stuck together, if you will. And so when I do that, I can then fragment it and break it apart. So now when I break it apart, the section that was bound to the protein, the DNA section that's bound to the protein, is stuck to the protein, and everything else just kind of falls off, right? Then I make an antibody that specifically binds to the protein that I'm interested in, right? And when I do that, I get these, right? And then I can use that antibody precipitation, right, to pull out the section of DNA I'm interested in. So I'm purifying the section of DNA that was attached to this DNA binding protein. And then I can just run it through next generation sequencing, and I can generate reads on it. I can sequence it, right? And then I can also map it to a reference genome, and I can see where it's located in the genome, actually. Like, I can actually literally look at where it's located. And I can see, oh, what other genes is it around, what genes is it regulating, and stuff like that. But, yeah, that's actually the end of this video. I hope it was helpful, you guys. And, yeah, happy studying. Have a good one.